or you can submit your video to Beecha by emailing her at marketing@pine.bio. Okay, so today I wanted to review what we are going to cover today. So we are going to continue in our week 2. We covered research literature review and project design. And hopefully some of you are starting to think about your project ideas and filling out that form. We are just completing the guide if you want a reference that you can follow. And we're going to be emailing you about that as well as posting it on the forum. So then yesterday we talked about genomics in virology and infectious diseases. So we talked about a few different aspects of genomics. Suparno is asking for the form. So let me just quickly show you where the form is. So we have this program, Genomics in the Virtual Lab. And once you get here, you go to the forum. So can you tell me if you see this forum, Suparno? Okay, when you get to the forum, you have here project submission. Okay, in the project submission, it's a sticky post. That means it's going to be yellow on top. You should see the form. And as I mentioned, this is a dynamic form. So your information is going to be visible to you. So in other words, if I log in, I see my information. If you log in, you should see your information. Okay, can you check whether you see the form and let us know? Okay, has anyone else seen the form? Does everyone else know where the form is? I know that some of you have submitted projects. Okay, great. In other programs. All right, excellent. So the goal is to fill out this form to get some feedback, and then you can continue and refine your project idea, introduce more detail, answer some gaps. If we discover some gaps and we point those out, you can address those, and this form is going to be dynamic. We will see the whole history of your project submission over time. So don't worry if it doesn't look like the complete thing yet. You can go back and you can edit and you can resubmit. So this week we are talking about next generation sequencing and how it applies to different domains. We're going to talk about the viruses and infectious diseases. We're going to talk about the fascinating world of the microbiome today. We'll do some hands-on tomorrow. We'll look at NCBI virus and we'll see how we can find interesting sequences and start comparing them. And you also have a number of different practical examples that we will be discussing. For example, last time we discussed the Spotoptera frugiperta SF9 cell lines. So this week, we will try to find the links between the sequence changes like mutations or other types of changes, abundance, variation, with particular phenotypes. Those phenotypes could be diseases or they could be conditions like anxiety. So we'll talk about that today. Again, the project submission, you post your submission here, and we haven't really updated some of your top profiles, but I've seen that Suparno has completed an introduction to metagenomics, and also Emeka was finishing transcriptomics 1. So this is really encouraging because I think if you look overall at the schedule that we have, you will still have some time to complete some of these projects, but if you regularly review the material, it builds one on top of the other. So what we will be covering today will incorporate some of the information that hopefully you had time to look at in Introduction to Metagenomics, and some of it in Metagenomics 1 and 2, so you can continue working on those after today's session. Let's review briefly what is it that we covered in our last session. We talked about genomics and virology, how to identify viral species and strains, how to compare analysis of do comparative analysis of different genomes from viruses, and look at viral diversity and mutations that are associated with disease pathology. Obviously, we did not have time to discuss each one of these topics in detail, but I hope that it gave you a glimpse of how genomics is very useful and actually transformative to the domain of virology and everything that has to do with infectious diseases. We talked about the virome. So who can tell me the difference between the virome and an infectious disease caused by a particular virus? So what do we mean? Okay. Yep. Exactly. Great. So the virome is all of the viruses. For example, the human virome are all of the viruses that live inside and on the human body. Not all viruses are infectious. Only a few are infectious. Okay. So when we think about the virome, it's important that the relationship between viruses and the environment that they live in, for example, other viruses, other bacteria, the host that they're in, plays a major role in how they function and what they cause. And so when we study the virome, we are trying to study and capture that diversity that we observe in the real environment. When we extract one single virus and we start focusing on that virus, for example, passaging it in tissue culture or cell culture, looking at it from an individual genome perspective, we get a very different view of what that virus actually does. So the virome includes many different types of viral elements. They include the eukaryotic virome, endogenous viral elements that actually are a part of the human genome, and prokaryotic virome, right? So the prokaryotic virome are viruses that infect bacteria. They are harmless to the human body, and they are keeping the bacteria and the viruses are in a certain balance when we have this particular condition. And we typically think of the virome as a part of the bigger picture, the microbiome. So today we'll talk about the microbiome. Now, when we look at those specific viruses, we can look at the diversity, so taxonomic classification. We'll talk a little bit more about that in terms of bacterial taxonomic classification, or we can look at a particular virus. So we can actually sequence a particular extraction from that virome and look at the whole genome or look at the changes in the virome. So when we look at those changes by comparing a genome to a reference genome, we're most interested in changes. So what kind of changes are there? There are mutations like substitutions, insertions, and deletions. Some of them have a higher confidence. Some of them have a lower confidence. What is the big factor between high confidence mutations or SNPs and low confidence mutations and SNPs? What aspect of sequencing allows us to have higher or lower confidence in mutations that we observe in the sequencing data? Okay, hint, it's right here on the screen. So what characteristic of sequencing allows us to determine mutations with higher confidence? So what plays a role in the confidence level in a specific mutation, right? Yes, and so what is the, okay, so for example, here we have a higher confidence, here we have a lower confidence, right? We don't know which one it is. Exactly, okay, so Parno got it correct, and Emeka, excellent coverage, right? So the higher the coverage, the higher confidence we might have about a particular change. If we have a lot of reads that map onto the same location, we have higher confidence in what we find, okay? So it's important to remember coverage plays a big role, and when we do sequencing, take a look at the coverage of that sequencing. All right, we also talked about different file formats. So we talked about FASTQ and FASTA and GTF. So who remembers what is the difference between FASTQ and FASTA? What information does one of them contain that the other one doesn't? Okay, so it has the quality score, FASTQ short reads and FASTA reference sequences. FASTQ is for small sequences. Right, so it's not just for small sequences, these are short reads, and those reads do contain an additional level of information, which is called the quality score, because we have a specific quality of each individual nucleotide. So typically FASTQ files are used to capture information about reads, which is the raw data coming out of the sequencer, and FASTA is an assembled sequence of the full reference genome. Yep, reference genome and its continuous sequence. Excellent. Okay, what is in the GTF file? Who remembers what's in the GTF file? What information does it contain? Okay, excellent. Annotation and specific annotation about genomic elements. For example, genes, exons, transcripts. Where do they begin? Where do they end? So we also talked about SAM and BAM files. Remember, we talked about SAM and BAM files. So SAM and BAM files contain information about the beginning and the end of each aligned read. So this is a result of alignment, and after alignment, we can have a SAM or a BAM file. They contain exactly the same information. One of them is readable by a human, like this one is a BAM file. If you look at the SAM file, it has the same information, but only the computer can read it. It's an ASCII code, so it only has code, a binary code that we cannot see. So then we also talked about the alignment of reads to the reference genome, how we can visualize those reads, and we talked about identification of mutations or specific changes. How do we actually visually see them? And we talked about the chromosome and the specific position. Remember, so here, if you did your Genomics 1 course, you will see that you can actually search for a specific gene or a position, navigate to there from there, and we can understand what exactly are these different types of changes. Okay? So these are called single nucleotide polymorphisms. Typically, we call them single nucleotide variants, polymorphisms, mutations. They're different terms, and they are different. They are used differently in different contexts. So when we talk about mutations, when we talk about variants or polymorphisms, it's also interesting to think what was the specific context. Were we talking about somatic mutations, like in cancer, or are we talking about mutations that are prevalent in a specific virus or a viral strain? Or are we talking about polymorphisms? Typically refers to variation that has a non-determined change. So instead of, let's say here we see G or A turning into a G, and in another person, maybe it's an A turning into a T. So that is a polymorphism. It can change to a lot of different things, but it's particularly in this position. When we talk about mutations, those are typically a mutation that is a change to this letter. and that letter is in the same position, and maybe it is always associated with a particular phenotype. So always think about these different characterizations. All right, so we had an assignment. Did anyone start looking at the project, the SF9, spotted tear, frugiperda, and rhabdovirus? Not yet. Okay, please take a look at that. That will give you an insight about extracting useful information from the non-mapped reads in a transcriptomic. Okay, great, Suparno. All right, excellent. So then we had a lot of genomic resources for virology, research, and education. So please take a look at that blog post. It's a blog post that kind of covers a lot of these virology resources where you can learn about different viruses and access data directly from these viruses. And also introduction to metagenomics. So did anyone, I know that Suparno did introduction to metagenomics. Did anyone else complete introduction to metagenomics? Okay, not yet. So hopefully you'll get a chance to take a look at those. So, but today we will cover some of the information that is reviewed in this course. Any questions about what we covered last time? When looking at the mutations listed on UniProt for my weekend project, the sequence didn't match the sequence of the molecule in PyMol. Why is that? So it could be for a variety of reasons. For example, in some cases where you have the full sequence of the protein, you might not have the full sequence present in the actual 3D model. So the 3D model might have a lot of missing elements from it, and that's why the sequence is different. Sometimes there's variation. So for example, I don't know if you were looking at the exact same query that you imported into PyMol. So when you look at those differences and you want to report them, for example, in a question like this, you're trying to understand why does it look, the sequence and the 3D model or whatever the 3D model sequence looks like, why are they so different? You might want to identify the specific sequences. Look at general characteristics like length. Is one shorter than the other? Look at similar portions. Can I find a similar sequence in both of them? Maybe they are shifted. Maybe it has multiple chains. Maybe the chains are different length. And maybe you can find one and the same sequence in both of those places. So it's definitely worthwhile to investigate. But if you want a clear-cut answer, I think an example would be best. For example, take a screenshot of the sequence, take a screenshot of your 3D model, kind of play around with it, see what you can find. Okay? All right, any other questions? Okay. Today in this session, we'll talk about the gut microbiome and microbial communities in general. So we'll talk about the bacterial genomes and the 16S ribosomal RNA, microbial diversity and composition, and we'll talk about analysis of 16S ribosomal RNA amplicon sequences data. So I hope that the outcome of today's session is for you to learn and understand how metagenomics can be studied directly from samples without culturing them in the lab. Metagenomics has transformed the study of microbial communities. What is the difference between metagenomics and, let's say, other types of omics? So typically when we talk about genomics, transcriptomics, proteomics, we're talking about a single organism, right? So genomics means the DNA of that particular host or that particular person or an animal. And the same with all of these other omics types, they focus on a single organism. When we talk about metagenomics, we're talking about a whole community and the diversity of that community, as well as how the community interacts with itself or with the host. And metagenomics is in a way a real transformational technology. Next generation sequencing has allowed us to study metagenomic and microbiome communities without isolating those specific bacteria in the lab. So how was microbiology kind of conducted in the classical sense? If you wanted to understand what an organism, again, a single bacteria, looked like, or what phenotype it caused, or how it behaved in different conditions, you had to grow it in a lab and then study that single organism. The problem is, as we've already mentioned with the viruses, is that these are actually a whole community, and the balance between the abundances of different members in that community is a huge part of that community. So in fact, we'll look at how changes in the specific communities inside the human body or inside a mouse's body can have a profound effect on some of the phenotypes that you might not even be thinking about, you might not be really aware of. So metagenomic data allows us to study those communities as a whole, as well as eventually to study individual members of those communities, but in the context of that whole community. Now we have these 3 courses. They're a little bit different here displayed on the screen from what you'll find on the Science Coach website, but nevertheless, they cover the same exact topics. Introduction to Metagenomics will introduce you to the general overview of different terminology that you have to know before you jump into metagenomics. Metagenomics 1 will talk about metagenomic data from some of the human microbial studies. How do we annotate and how do we find specific types of bacteria in our metagenomic data? And Metagenomics 2 will focus exclusively on the 16S ribosomal RNA, which is something that we'll talk about today. So for those of you that did take the course, I want to ask you a few questions. Which of the following terms is unrelated to 16S ribosomal RNA? Okay, so Soparno just completed this course, so he remembers it's number 5. Excellent, Soparno. Anyone else? We'll talk about these terms in today's session. So if you still don't know what is amplicon or next generation sequencing, phylogeny, hypervariable, yes, or protoplasm, we will talk about some of these things. Okay, and Mecca also. What about this one? Taxonomic classification can best be described as? Okay, 16S RNA is used in phylogeny because of the low rate of mutations, correct? So that is something that we'll talk about just in a few slides. So I don't want to give the answer right now, but yeah, we'll learn about 16S ribosomal RNA. They have variable rates of mutations across that whole gene. Soparno Das is saying number 3. Muiwa is also saying 3. So let's see. Studies of increased rates of disease within a collection of microorganisms, so taxonomic classification. You should know this because we already talked about taxonomy. New ways of simultaneously sequencing multiple genomes, studying the extent to which genetic sequences diverge, a way of preparing genomic material to be sequenced, the removal of variable regions of RNA to increase experimental validity. Okay, so yes, most of you got number 3. That is correct. So we'll talk about how can we study genomic material. We look at by divergence or difference. And so we will talk about how that can help us organize the data in some kind of a logical way. And the last question, shotgun sequencing is an important technique in metagenomic studies. Which of the following are true regarding this type of sequencing? So number 1, shotgun sequencing is based on multiple reads. 2, utilizes a reference genome that is compared to the sequence reads. 3, can be performed on DNA and RNA, and all of the above. All right, excellent. That is correct. So it looks like you all have a head start. Now in Metagenomics 1, there is a project example. That project example is from American Gut. American Gut is a citizen science project that allows scientists to collect samples from around the country first, and then it was expanded around the whole world, and to study self-reported phenotypes associated with microbiome of those individuals. You provide a sample, any one of you can do this. You provide a sample, you get a kit, you have to pay for them to do the sequencing, but then your data is associated with everybody else's data, and you can see how you compare to everybody else. You can see here, for example, different ages and populations, different body sites, and the American gut population based on a particular type of bacterial species. And you can also see that, for example, you will get a report. So this is how your sample compares to everybody else's. So you can understand how normal you are or how comparable you are to other people your age, etc. This is a very interesting citizen science project because it leverages the citizen, so us, that contribute samples to a real scientific study, and scientists are able to infer kind of what is normal at a very detailed level. So in that project, in that example used in Metagenomics 1, what we do is we actually build a pipeline, and that pipeline allows us to understand the abundance of different bacteria based on a reference database. And that reference database organizes the 16S ribosomal RNA into a table of abundance. This is a table of abundance. You can see that the table of abundance from metagenomic studies, as opposed to, let's say, gene expression studies, have a lot of zeros. In terms of data, this is called sparse data. It has a lot of kind of zero information, zero inflated data. And as a result of looking at the different levels of taxonomy for these bacterial species, you can then infer difference. So that is a good starting point for anyone interested in metagenomics. It's a fascinating field that has been really making a lot of progress over the past several decades to discover how the microbiome is actually affecting many, many aspects of human health, including many different types of diseases, many non-clinical conditions, psychological conditions, and different sensitivities to different foods and different types of conditions, environmental conditions, and also even treatment. So even microbiome is important for the treatment because obviously the microbiome inside the gut has a role in how we are able to process different foods, process different chemicals, and effectively convert them into active elements that then are used for treatment. So a very important area of study. This is a very good place to start and understand what goes into the sequencing and annotation and analysis of this kind of data. Okay, so any questions about metagenomics as we kind of get started? I want to focus more on 16S ribosomal RNA in the rest of today's session. So just want to make sure that some of these were clear to everyone. So I know that only some of you have answered different questions. Maybe for the rest of you, this was a little bit confusing. Do you have any questions about what we just went through that you would like me to expand on? Okay, all right, so who knows what this is? Okay. Okay, tree, evolution tree, phylogenetic tree. Any other ideas? Evolution tree, phylogenetic tree. Okay, so this is actually, you're all kind of correct. It's the phylogenetic relationships of these different organisms. What this seeks to represent is the tree of life, or a universal tree of life. And really, it's a metaphor. The tree of life is a metaphor or a model, or it's a tool, a research tool that is used to explore the evolution of life and describe the relationships between organisms that are both living and extinct. And it was first described by Charles Darwin. So whenever he wrote about this, and this is actually his sketch of the tree of life. This is the first sketch of the tree of life. It was found in one of his notebooks in 1837. And the tips of the branches show the species that are still alive today. The tree also shows those that are now extinct. What he explained was that the first growth of the tree, many a limb and branch has decayed and dropped off, and these fallen branches of various sizes may represent those whole orders, families, and genera which have now no living representatives and which are known to us only in a fossil state. Orders, families, and genera are all groups that can be used to classify organisms. And so this sketch was around 1837 that was first produced, and it became known as the tree of life or an evolutionary tree. And you can notice right here, it says, I think, right? So it was a model to organize the thoughts about the relationships of the different species. And these species are based on evolution. So we call them evolutionary relationships. For example, a recent version of the tree of life would show a line between some types of dinosaurs and the earliest birds, as scientists reason that birds evolved from a particular lineage of dinosaurs. This means that species that are closely related are found close together, stemming from the same branch. For example, humans, chimpanzees, gorillas, and orangutans are all great apes, so they all belong at the same branch of the tree of life. So let's take a look. Here you can see the more modern version of this tree, right? So we see here different chimp, lemur, golden lion, tamarin, and the human, right? They are all apes, and they all fall under primates, etc., right? So what is this tree? So this was kind of a very basic idea. So this is a more expanded version of the same tree of life diagram that eventually Darwin put together. And so this concept became the basis for his theory of evolution. And this is in the chapter 4 that he presented an abstract diagram of a theoretical tree of life for species of an unnamed large genus. On the horizontal baseline, hypothetical species within this genus are labeled A through L and are spaced irregularly to indicate how distinct they are from each other, right? And here you have phenotypes, right? So that is really where the term phenotype comes. So the phenotype are the differences between these different species, and the more similar they are, the probable origins are more closely related to them. Okay, so here you can see in Darwin's own words, thus the small differences distinguishing varieties of the same species will steadily tend to increase till they come to equal the greater differences between species of the same genus or even of distinct genera. So this is a branching pattern with no names, right? And we kind of see this order of lower to higher species. Okay, and this is what Darwin said himself. The affinities of all the beings of the same class have sometimes been represented by a great tree. I believe this simple example largely speaks the truth. The green and the budding twigs may represent existing species, and those produced during each former year may represent the long succession of extinct species. At each period of growth, all of the growing twigs have tried to branches out on all sides and to overtop and kill the surrounding twigs and branches in the same manner as species and groups of species have tried to overmaster other species in the great battle for life. Okay, so this, I think, gives us a nice example of how to visually think of this tree of life, of the evolution, and to understand how we measure the relationship. We can look at homology or similarity. So for example, here you can see different properties of these organisms and how the relationships between them are defined by these specific properties. So for example, here you have vertebral column, right? So this is the basic phenotype. They all possess a vertebral column, right? Except for the 0, right? So right here, it did not exist, and it appeared right here. So therefore, this is a branch. Then we say hinged jaws, right? So again, if we move forward, and 4 walking legs, etc. We can see this as an example, the relationship between specific bones, right? So here we have different structures, and we derive these structures from a common ancestor, and they're called homologous structures. And these are homology of skeletal components of vertebrates. So again, shared homologies allow us to devise hypothetical family trees of living organisms called phylogeny. So that's where the term phylogeny comes from. These can be represented as a branching diagram called a phylogenetic tree or evolutionary tree, as you correctly identified. Now, how does this relate to sequence information? On a genetic level, it means that species contain the same or similar DNA sequence, genes, and proteins. Structures as well as proteins are thought to have the same or similar function across a variety of species. This similarity is typically thought to exist because of a shared common ancestor. While homologs are defined as very similar to each other, it is important to note that this does not mean that they're exactly the same or allow for the same exact function to be carried out in the body. As an example, here you can see the Hox gene. Yeah, so this is the different Hox genes. Hox genes controlling body segment development, which are, so the Hox genes are actually a subset of homeobox genes, and they're a group of related genes that specify regions of the body plan of an embryo along the head-tail axis of animals. Hox proteins encode and specify the characteristics of position, ensuring that the correct structures form in the correct places of the body. For example, Hox genes in insects specify which appendages form on a segment, for example, legs, antennae, and the wings in the fruit flies. And Hox genes in vertebrates specify the types and shape of vertebrae that will form. In segmented animals, Hox proteins thus confer segmental or positional identity, but do not form the actual segments themselves, right? So it is a gene that controls development and causes different types of segments to appear first, second, third, etc. Studies on the Hox genes in ciliated larvae have shown that they are only expressed in future adult tissues. In larvae with gradual metamorphosis, the Hox genes are activated in tissues of the larval body, generally in the trunk region, that will be maintained through metamorphosis. In larvae with complete metamorphosis, the Hox genes are mainly expressed in juvenile rudiments and are absent in the transient larval tissues. So this is an example of a gene, and we can see how the gene is actually related to the phenotype. And so the mutation in these genes causes homeotic transformations. Homeotic transformations were first identified and studied by William Bateson in 1894, who coined the term homeosis. After the rediscovery of Mendel's genetic principles, Bateson and others realized that some examples of homeosis in floral organs and animal skeletons could be attributed to variation in genes. So that's where the relationship between these mutations and the function of genes helps us associate appearance of mutations with changes in phenotype. Okay, so thus when we have a phylogenetic tree, we have a sequence of relationships between different observed species, which is what we see at the ends of these branches. They are the observed species. A group of those which form on a specific branch is called a clade. So that clade could be either the small clade or the bigger clade, and it is defined by nodes that branch out from these branches. So the origin and the end result could then be measured in evolutionary time, right? And so that's a property of the phylogenetic tree. We can have an evolutionary time, which is measured in some cases by appearance of mutations. So the number of mutations from the species that we observe to the node describes the distance. If we have a real time, so for example, we can take a sample from here, and we can take a sample from here and sample from there, we can associate the rate of mutation with real time. So we can link evolutionary time with real time. Now, sometimes these form, just the shape of these trees are, presented in different formats. For example, this is a radial phylogenetic tree, but it has the exact same information, just organized in such a way that we can more easily see the whole structure at the same time. Okay, so where in this more modern version are the microbes? Who knows? So where are we? Let's say, where are we? Where are humans in this tree? Okay, yeah, eukaryota, right? So we are right there with the animals. All right, great. And what is interesting about this relation, right? Where are the bacteria or the microbiome? They're right here, bacteria, right? So these are the three main early branches of the tree of life. And they have been intensively studied by microbiologists because the first organisms were microorganisms. Microbiologists, led by Carl Weiss, have introduced the term domain for the three main branches of this tree, where domain is a phylogenetic term similar in meaning to biological kingdom. To reconstruct this tree of life, the gene sequence encoding the small subunit of ribosomal RNA, which is also called SSU of rRNA, or the 16S rRNA, ribosomal RNA, has proven very useful. And the tree, as shown in this picture, relies heavily on information from this one single gene. These three domains of life represent the main evolutionary lineages of early cellular life and currently include bacteria, archaea, which are single-celled organisms superficially similar to bacteria, and eukarya. Eukarya includes only organisms having a well-defined nucleus, such as fungi, protists, and all organisms in the plant and animal kingdoms. The gene most commonly used to construct this phylogenetic tree and the relationships between the microorganisms and the small subunit ribosomal RNA gene. And it is because its sequence tends to be conserved among members with close phylogenetic distances, yet variable enough that differences can be actually measured. So the SSU rRNA as a measure of evolutionary distances was pioneered by Carl Weiss when formulating the first modern tree of life, and his results led him to propose the archaea as a third domain of life. However, recently it has been argued that SSU ribosomal RNA genes can also be horizontally transferred. Although this may be rare, this possibility is forcing scrutiny of the validity of phylogenetic trees based on SSU ribosomal RNA. Okay, so what is this gene? Here we have the prokaryotic and the eukaryotic ribosomal RNA. Okay, you see that there is a top or the large subunit and the bottom or the small subunit ribosomal RNA. And this is the 16S ribosomal RNA, which is encoded by, so this is the gene that actually encodes for this small subunit. In the humans and eukaryotes, this is the 18S ribosomal RNA. So this is the component of the whole ribosome that binds to the Shine-Dalgarno sequence. The genes coding for it are referred to as 16S ribosomal RNA gene and are used in reconstructing phylogenies due to the slow rates of evolution of this region of the gene. And we see that multiple sequences of the 16S ribosomal RNA gene can exist within a single bacterium. So this gene contains a number of different regions. It is approximately 1,500 base pairs long, the whole gene, and it has 9 different conserved regions. Each region is between 30 to 100 base pairs long, and full hypervariable regions can be assembled from a single Illumina run, right? Why is this important? When we have these different variable regions, if we look at the full 16S ribosomal RNA, okay, based on what you know about next generation sequencing, is this too long or long enough for the regular read, right? What is the length of the read typically of next generation sequencing? How long is a single read typically? Okay, 160, but what is the range? Let's give it a range. $30 to $3,000. It's more like 30 to 300 actually. And this, as you can see right here, that we have 30, okay, 50 to 300 great Soparno. So we have each region is up to 100. So you can actually sequence the full hypervariable region. So these are called variable, hypervariable regions, right? So in the whole sequence overall is fairly conserved, but this specific region, so you can see that they are on the edges of this structure. This is the 2D structure. They are more variable, meaning that if we capture one of these sequences, we want to have a very high quality sequence associated with that particular portion. So the bacterial 16S gene contains 9 hypervariable regions ranging from about 30 to 100 base pairs that are involved in the secondary structure of the small ribosomal subunit. The degree of conservation varies widely between hypervariable regions, with more conserved regions correlating to higher level taxonomy and less conserved regions to lower levels, such as genus and species. While the entire 16S sequence allows for comparison of all hypervariable regions at approximately 1,500 base pairs long, it can be prohibitively expensive for studies seeking to identify or characterize diverse bacterial communities. These studies commonly utilize the Illumina platform, which produces reads at rates 50-fold and 12,000-fold less expensive than 454 pyrosequencing and Sanger sequencing, respectively. So it is 12,000 less expensive than Sanger sequencing, right? So that makes it much more affordable and much more available for people. So while cheaper and allowing for deeper community coverage, remember the key is coverage, Illumina sequencing only produces reads that are anywhere between 30 to 300 base pairs long. And the Illumina MySeq is a specific instrument that actually allows for that 300 base pairs and has no established protocol for reliably assembling the full gene in community samples. So as a result, this is the typical process. We have a sample collection, DNA and RNA is isolated, and then specifically a region, a particular hypervariable region is amplified. In many, many studies, it's going to be the V4 hypervariable region. And when we say it is amplified, it means that using PCR primers, those are actually reproduced many times. And then based on the variability, we can analyze the data to understand the composition. So here is that hypervariable region. Okay, you can see that this is the specific way to actually amplify using PCR. You have a specific primer for the forward and for the reverse strand of that sequencing. Illumina sequencing, yes, comes under next generation sequencing. So Sanger is typically referred to as first generation sequencing. And then 454 and Illumina are next generation sequencing. So Perimala, if again you can make those public. So V1 to V9 are called hypervariable regions. So the overall gene, the 16S gene, which is 1,500 nucleotides long, is more conserved, but the specific regions are actually hypervariable. So they have a higher variability. Let's take a look. I actually have a slide here. So this is a slide about those hypervariable regions. So this is frequency of most common residue in 50 base window. Okay, so this is a measure of conservation. Okay, and this is the base position, right? So the whole gene is 1,500 base pairs. And then you can see these are the specific hypervariable regions. Okay, remember what information does this conservation give us? How does it relate to taxonomy? Who can tell me how does conservation relate to taxonomy? Okay, so closer or more, so closer, remember that taxonomy can be measured at several different levels. Okay, for example, you have the whole kingdom, right, is a big group that contains a whole lot of variability inside. Closer in relationship when variation are lower, we can identify the variability of sequences, which can help us identify relations among different species, right? So species and families and genera, these are all levels of taxonomy. And the lower variability, so let's say overall, this gene is conserved as compared to others, right? So if you have more diversity in a group, that means you have more groups within that group, right? And then if you have less diversity, that means you have less groups. So if you start organizing them, I have one big group, then within that group, I have just a few subgroups, and then each one of those has a few more, and each one of those, you end up with a tree, right? So it's like big group, then lower level group, lower, lower, lower. So the higher the diversity, or the less diverse they are, less conserved. Okay, the more conserved the reads, the more related they are, right? Between organisms. So if they are more conserved among many organisms, that means that they are the same amongst many organisms, and that puts them in a higher level taxonomy. If they are more diverse between organisms, that puts them at a lower level of taxonomy. So here you can see more conserved, right? So this is this allows us to study those specific levels of taxonomy. Okay, so how do we actually infer this level of taxonomy? We use what's called an operational taxonomic unit, or OTU, which is an operational definition that is used to classify groups of closely related individuals. So for example, you can think of hierarchical clustering, right? Hierarchical clustering can be used to organize these OTUs into a particular group. Okay, if I have a number of sequences, okay, you can see that OTU1 has some level of diversity. They are almost the same, but they're just a little bit different, maybe one nucleotide difference. Okay, OTU2 has just a little bit of differences. So these differences make them into one single OTU. So as a result, we can sequence these regions and get those sequences organized in a single sample, right? So that single sample is characterized by differences between these operational taxonomic units, because we don't want to look at each individual variation, each mutation. So we kind of group them by similar number of mutations so that they form some kinds of groups. Okay, and then those groups, the records of those individual sequences are organized in different databases. So what are some of these databases? SILVA, RDP, which stands for Ribosomal Database Project, right? So people that actually annotate these sequences when they appear in real life. And here you can see a reference publication which refers to the comparison between these different repositories of known sequences, NCBI, RDP, SILVA, Green Genes, etc. And what they have found is that actually SILVA is one of the more comprehensive resources that is comprehensive but also quality checked and regularly updated. Okay, so we have the process of sequencing. The sequencing information yields some kind of a sequence that is known and is organized into a group. We call those groups operational taxonomic units. And based on operational taxonomic units, we can study diversity and how it relates to that taxonomy, to that phylogenetic or evolutionary relationship. So for example, in the American gut, that is what they are looking for. They're not looking at the full sequence of all of the microbial genomes present in a sample. They're actually looking at that example of the 16S ribosomal RNA and the diversity, okay? And also presence or absence of specific groups of bacteria in a sample. So let's look at another project. This project is looking at the relationship between the gut and the brain. Okay, so how can the gut actually be influencing the brain? And one of the studies that we're going to look at today is about this gut-brain axis, and it looks at mice that have different types of diets and their behavior is measured. Okay, so what was the experiment? The mice were given two types of diet, high-fat diet and regular diet, which is called chow. So high-fat diet will be HFD and chow will be standard diet. So the mice given HFD microbiota, meaning that they got the microbiome extracted from high-fat diet mice and they were implanted, that microbiome was implanted into these mice. They were found to have significant and selective disruptions in exploratory, cognitive, and stereotypical behavior compared with mice with control diet microbiota in the absence of significant differences in body weight. Sequencing-based phylogenetic analysis confirmed the presence of distinct core microbiota between groups with alterations in alpha and beta diversity, modulation in taxonomic distribution, and statistically significant alterations to metabolically active taxa. High-fat diet microbiota also disrupted markers of intestinal barrier function, increased circulating endotoxin, and increased lymphocyte expression of ionized calcium-binding adapter molecule 1, toll-like receptor 2, and toll-like receptor 4. Finally, evaluation of brain homogenates revealed that that high-fat diet-shaped microbiota increased neuroinflammation and disrupted cerebrovascular homeostasis. So what you see is this untrivial relationship between simply the bacterial community, the microbiome or the microbiota inside these mice associated with the mouse behavior and different types of exploratory cognitive behavior, right? Observing a disruption there. So what we see, how did we generate, or how did the authors of the paper actually generate this type of data? They took 8-week-old male mice and separated them into two groups. One group was fed for 10 weeks high-fat diet, and another one for 10 weeks was given chow, regular diet. Okay, then their microbiota was sequenced. Then those mice, or new mice, were taken. Those are 3-month-old male mice. And after a reduction in their regular microbiome, their natural microbiota, they were given this behavioral test, okay? And so after they were studied and we understand what kind of microbiome they have, they actually were implanted this microbiome, the high-fat diet or the chow microbiome, and then they were again behaviorally tested and sampled, and so we can study the results of their microbiome. Okay, so does everyone understand the experimental design and how this data was generated? Okay, are there any questions about how the data was generated? Okay, now also remember that the microbiome or metagenomic sequencing could be done from different places. You can take it directly from the gut, which is called cecal, and then you can take it from the fecal samples as well, which is called fecal samples. Okay, let me repeat. So they had donor mice. So the donor mice were on different diets, a high-fat diet and a regular diet, and they gave their microbiome, right? So after being fed for a particular diet, they only extracted their bacteria from their gut, okay? So they took their microbiota. Then they took new mice, they kind of lowered or reduced their bacterial load. So they kind of cleared their regular microbiome. What antibiotics do is they actually lower the level of diversity, right? And they kill a bunch of these bacteria. So they were given this antibiotic and then implanted this new microbiome. For example, given, you know, portions of that microbiome with food or somehow implanted like that. And after that, they were studied, right? So they were studied before and after to observe how their behavior actually changes. And so now you have two groups of samples. Some are high-fat diet, right? So they were recipients of the high-fat diet microbiome, and they were studied for what was their behavior like. Okay, and we were trying to associate changes between behavior and microbiome changes. So importantly, their weight was not changed, right? So they all are the same weight. Does that make sense, Muiwa? Or anyone else? Does, you know, anyone else have any questions about this? So the 3-month-old male mice were given antibiotic to reduce bacterial load. They were separated into two groups. Yep, samples taken. Well, the difference now will be not in the diet, but in the microbiome, right? So they got different types of microbiome, which was a product of different diets. But it's not like they were mice that were fed different diets. And so we can associate the behavior changes with diet. We're associating behavior changes with the difference in microbiome. Okay, Muyo, does that make sense? Not the same. No, the microbiome is different, right? So the microbiome actually comes from here. So you had donor mice. The donor mice were on different diets, and their microbiome was taken. Okay, so their microbiome, so think of it, we're trying to differentiate here between the diet itself and the microbiome. Okay, yeah, so I think a very interesting thing, right? So now their microbiome was taken, implanted into these recipient mice. Why is the bacterial load being reduced? Is it to isolate population of microorganisms which originate from a specific diet provided to them? Well, you know, you want to implant a new microbiome. So if you just add this microbiome to existing microbiome, you don't know what you're going to get. You're going to get some kind of a mix. But you first reduce it, so you kind of cleanse it, right? Remove previous microbiome, and then give them a new microbiome, and that new microbiome establishes itself in this kind of a void, you know, of lack of diversity and kind of reduced load. Okay, so any other questions about the design? Okay, clear to everyone, right? Okay. Yes, all right, great. Okay, so now let's actually do the analysis, okay? We're not going to actually do the analysis and run the analysis. We're just going to look at a demo right now, but all of this is described in metagenomics too. So if you want to actually reproduce this analysis and see where these files are and what are the sequencing files, etc., you can actually find them in metagenomics too. Okay, so why is the load being reduced in case of chow? Isn't that the regular diet? Yes, it is, but you want to have a, you know, this is an experimental control, right? Right, so exactly. So you're making a control. So if maybe it's just because of the stress of, you know, reducing the microbiome load and introducing new microbiome. So to eliminate that factor of stress, you're saying, let's do the exact same procedure and we'll just observe the resulting behavior. So it's not associated with anything else but the specific microbiome composition. All right, so let's take a look at this pipeline together. This is called the data to pipeline. Okay, so this pipeline is available. You can run it in R, but we're just going to do the demo so that we understand the logic of this pipeline. What does this pipeline actually do? So let's go to the server site. Okay, so this is the link. Let me know if once everybody gets there. So let me know once you're here. All right, excellent. So once you're here, you scroll down all the way to the end. This is the amplicon 16S or 18S. Okay, and here you have the demo metagenomics amplicon gut-brain axis. So click here, and we'll walk through each step to understand what that step means. Okay, so the first one is you click on start, and you already remember what is the 16S ribosomal RNA and why are we looking specifically at the hypervariable regions. Step before that, so again, you're here, you click on the drop down arrow and you go to the demo metagenomics amplicon. Okay, so, all right. Okay, so if you click on start here, you will see how we already discussed this 16S ribosomal RNA and the specifically V4 hypervariable sequencing, right? And then we here have the hierarchy of biological classification. So our objective is to take in a number of samples. Let's take a look at what samples we actually have. Okay, so if all of you can download references chow HFD supplementary tables, so just click on this arrow, not on this one, on this one. Okay. So what you should see is the names of samples, BKF, so F here stands for fecal, and you can see that there's 3 for chow and 3 for high-fat diet, right? So we have 3 samples each. This one, the one that is closer to the word, so this one, okay? So this one will give you the actual file. This will give you a link to that file, okay? So let me know once everyone got this file. Okay, so this is an important file because if you ever want to run this kind of input, this is the input file. So if you ever want to run this kind of a pipeline, you need to annotate your samples in a way that the script knows which ones are which. All right, great. Okay, did everyone else get this file just to take a look at what's inside? All right, let's click on OK. All right, and here, sometimes, okay, I'm sorry to hear that, Kylie, but I'll also, you know, if you do want to do bioinformatics, having enough RAM and good storage space, etc., is going to be very important. So we're hoping to eliminate those requirements for the majority of you with, you know, being able to use a web platform. But of course, the web platform also requires some functionality. So you know, we'll see if we can provide you with an R script, and you can try and figure that out. But I think it will be a little bit more involved and complicated. Okay, so here, the first step after we load our samples, right here. So, okay, let me, by the way, anytime you want to go back, you just right click. Okay, so you click on Start, Input File, and then here, References, chow HFD, Supplementary Table. So this right here. So Muiwa, let me know once you've got this. And Kylie, are you using Chrome? Okay, do you know how to open the console in Chrome? I think it's F12, but you can also see it from the developer menu. So maybe you can look. Yes, click right here on this arrow right here, Muiwa. So Kylie, if you could email us a screenshot of what you see in the console. Okay? Yep, a TXT file. So Muiwa, this is what you should see. This is what the file should look like. You have the run. So this is the individual file, and this is what type of a file it is, associated with the chow diet or HFD diet. All right, so you click on OK. Sometimes you want to remove some, you know, known attached sequences to those reads, but in this case, we're going to go to QC filtering. So this is going to be the filtering of reads based on quality. Okay, so again, this just looks at individual sequences of those reads and looks for quality control. It actually generates a graphic that shows quality, and some of the low quality reads are just going to be removed completely. Okay, then this is, okay, yep. So then what happens is, so we've pre-processed the reads, we have removed some of our low quality reads, and now we need to make sure that they are paired reads, because one of the elements of MiSeq metagenomic sequencing is that this is paired end sequencing, provides us with higher confidence in the individual read sequences. And once we find that all of them have pairs, we know that those are actually high quality, good reads for us to use. Okay, so after reads are analyzed for quality and are trimmed in the same way, we need to eliminate reads that do not have a matched pair. This method outputs a dereplicated list of unique sequences and their abundances, as well as consensus positional quality scores for each unique sequence by taking the average or the mean of the positional qualities of the component reads. Okay, so we are using information from the FASTQ inputs from both reads, the forward and the reverse strand. Okay, then those pairs are merged and we can prepare the operational taxonomic units. Remember, those operational taxonomic units have very small variation within a group. So that unit might include small variations that are insignificant. That way we can reduce the number of individual types of units that we are reading in our whole data. Sometimes reads will include a starting point of one and the end point of another actual biological sequences, and those are called chimeric sequences. They combine elements from different biological sequence samples, and so we want to reduce those. You can see that the frequency of chimeric sequences varies substantially from dataset to dataset and depends on factors including experimental procedures and sample complexity. Here, chimeras make up about 21% of the merged sequence variants. But when we account for the abundance of those variants, we see they account for only 4% of the merged sequence reads. Yeah, chimeric sequences are sequences that come from two different biological sequences. So you have a technical sequence and you have the different source that is definitely present in that one sequence. Okay, so you can see chimeric sequences are identified if they can be exactly reconstructed by combining a left segment and a right segment from two or more abundant parent sequences. Okay, so you've got, you know, multiple OTUs that are more abundant, and then sometimes they kind of combine those two together. And that just introduces a lot of noise, right? And a lot of unanswered questions. And then so once we have these highly reliable OTU sequences, we can actually assign taxon. And those taxons are based on a database, okay? So it uses a database to look for taxonomy of these individual OTUs. Remember kind of what we did last time? We looked at annotation of specific sequences. Here we also use a database and we have a particular sequence of known OTUs. Okay, then once we are done, this is the data to pipeline. Okay, we want to also visualize the results. APE is just to visualize the phylogenetic tree. Okay, the phylogenetic tree of the different samples. How do they work together? And then we have a package called phyloseq, which actually looks at these operational taxonomic units and looks at how they are associated with a particular type of sample, right? So it kind of combines the information by group. And here are some of the outputs. Okay, so now we have our full pipeline. Click on end. At the end, we will see a number of different outputs and we'll talk about what they exactly mean. Okay, once you click on there, you will be able to see some of these pipeline output files, including an abundance table, an error plot, and the individual OTU sequences, okay, phyloseq plots, etc. So did everyone get to this point? All right, excellent. So for those of you that want to try this again at home, you will have this recording. You want to just follow the steps. It's fairly simple one by one. You just get to understand what each step actually does. So let's take a look at what these steps do and what is the actual output. So here we have the whole data to bioinformatics workflow. The particular focus of this workflow is to eliminate noise, because noise, when you have a lot of unknowns, it's easy to add noise to these unknowns that people want to study. And so you can see how some of these outputs are going to provide us denoising capabilities, and others will actually construct a taxonomy, but also we can look at individual OTUs that we can then explore. Okay, so here are some of the outputs that are going to be interesting for us. We see the abundance table. So what is the abundance table? It has different samples. Okay, this is many more samples than we have in this demo pipeline, but we will just have names of samples in the abundance table and OTUs. The less OTUs we have with zeros across, the better it is for us to analyze. Okay, and this number is the abundance of that OTU. For each OTU, we will also have taxonomic annotation. So this taxonomic annotation is by kingdom, phylum, class, order, family, genus, and species. Okay, so here you can see that for species and genus, many of these OTUs are actually unknown. Right, so in some cases, if we want to go to a lower level of taxonomy, so this is the lower level of taxonomy, this is the higher level of taxonomy, the lower we want to go, the more unknowns we will have. And this is just based on annotation. Okay, so then we can construct, these are abundance visualization. Okay, and let's see, what can you tell me based on this, these two charts? Okay, so how are these two diets different between these two conditions? Okay, the first thing that I will ask is, what level of taxonomy was used to produce these charts? What is the level of taxonomy? Okay, so Parno is saying family. Anyone else? What do you think? Okay, so let's go back and see, what do we have here? Okay, so first it's the kingdom, right? Then we have phylum, class, order, family, genus, species. Okay, so these are the examples that you have. So looking at these charts, okay, it actually says on these charts, what is the level of taxonomy? Okay, Sanjali actually correctly identified this is phylum, right? Why do we say phylum? Right here, it says phylum. So the abundance is colored by different colors, and they are colored by phylum, right? So where is phylum in the order of taxonomy? Phylum is a level of classification below kingdom and above class. Okay, so look over here, you have kingdom, and this is class. So this is right here, phylum. Okay, so phylum is not a very detailed view of what's going on. It's a pretty high level view. Suparno was saying family. Family is right here, right? It does have quite a number of available ones, but genus and species actually has too many NAs, right? So it doesn't make sense in a lot of these studies to use genus and species. It's better to go family or above. Phylum gives us a very general overview of what's going on. So let's take a look at what else we can learn from this chart. Okay, so this chart specifically has more samples, right? So look, this is a different type of number, but we'll learn about the differences between these samples. Okay, so look, we have different colors associated with different taxonomic order of phylum. And in this case, for the chow, for the regular diet, we have Bacteroidetes, Firmicutes, Proteobacteria, Tenericutes, and Microbia, right? Microbia, Microbia. So another important aspect of what you want to look for is that this contains a number of different samples, right? So here you can see that some of them are BKCM, which is cecal samples, and then BKF. F stands for fecal samples. So in fact, we have these BKFs and BKCM. In here, for example, this is very obvious for a high-fat diet, right? You have the BKCMs are on average higher than BKFs. Okay, so let's look at specific phyla that are different. They're different colors. So for example, Bacteroidetes here are red and here are orange color. Firmicutes here are green and here are a little bit of a different color of green, right? So if you look at the relationship, okay, this is what we know about the different sites where some of these bacteria are typically abundant. You can see different sites in the body, and here you can see different types of Bacteroidetes, Firmicutes, etc., proteobacteria that are associated with different sites in the body. Okay, so one way that we can think of these is where are they typically present in the body? Another way that we can start thinking about this is what is their function and abundance. So the more diversity we have, the more abundance of particular types of bacteria, the more we can study what they actually do, right? So if we look at the high-fat diet, what is the most abundant high-fat diet component that is not present in the chow? Which one do you see for a high-fat diet that is not present for chow in great abundance? Proteobacteria. Okay, so proteobacteria is right here. Proteobacteria is right here, right? So it is not very abundant here. It's not very abundant here, but it is present in some of them. Firmicutes, so this firmicutes and this has firmicutes. What does one of them have that the other one? Okay, so Saccharobacteria primala, that's correct. So Saccharobacteria is not even present here, right? Which means that in terms of this scale of abundance, it's indistinguishable from zero. Okay, and actinobacteria. So let's look up actinobacteria. What does actinobacteria actually mean? Okay, they're gram-positive bacteria with high guanine and cytosine content in their DNA, which could be, okay, they're unicellular. Let's see. Okay, we can look at maybe actinobacteria, gut microbiome. Okay, here, for example, in this article, you can see that actinobacteria and delta proteobacteria increased their respective proportions. Let's see. In the gut of both groups of animals fed with a high-fat diet, which was accompanied by a reduction in the abundance of class Bacterial, right? So for example, it has been also studied that actinobacteria is increased in a relationship to the high-fat diet. Okay, so this is something relevant that we are observing. The other one, as correctly pointed out, was Saccharobacteria. Saccharine. Okay, there's also some publications here. High-level dietary fiber upregulates colonic fermentation and relative abundance of saccharolytic bacteria within the human fecal microbiota in vitro, right? So we do have some insights here, right? Obviously, this requires a much more in-depth study, but it points to the fact that some of these might play a role, right? Let's look at another type of a comparison. Okay, this comparison is called alpha diversity and beta diversity. Okay, what does that mean? That means that one of them, we have a way to measure how diverse each one of these groups of samples is. Okay, so here you can see, this is from the actual demo pipeline that we did. So you can see the reduced number of samples, six total. And the green ones are diversity measure for Chow-1 is a method to measure this diversity, or the Shannon measure of diversity. You can see that they show fairly similar results, with this particular sample having a much higher alpha diversity measure than the rest. And overall, because of this sample, right, or because maybe of both of these samples, we can say that the high-fat diet, right, so let's see, which samples are these? Okay, so 13, 14, and 15 are all high-fat diet. So 13, 14, and 15 are high diet. They are higher alpha diversity. Okay, this is beta diversity, which looks to identify kind of like a principal component analysis, but called NMDS. So this is multidimensional scaling. MDS is multidimensional scaling. It shows us a separation between these two types, right? So it actually shows us a very clear difference in diversity. This is within a sample. This is a measurement of diversity within a sample. And this is a measure of diversity between all of our samples. Okay, that's why NMDS is kind of like principal component analysis. It shows us variability in our whole data set. So in our whole data set, we can see that there's a major difference in diversity between these different sets. So we have two ways of looking and comparing these microbial communities. One way is to look at abundance of particular taxonomic level. Another way is to actually look at diversity. So the more diverse something is, or the less diverse something is, does it actually tell us something about the groups of samples? Okay, and yes, it does. Groups of samples here are very different, right? In other words, we can just draw a simple line and separate completely two of these types of samples. Okay, do these charts make sense? Do you have any questions about these charts, what they actually show? Okay, so this is the beta diversity chart. Beta diversity is like looking at a way to measure differences in abundance, or sorry, in diversity overall, right, in your whole data set. So let's imagine we have different colors of Skittles, okay? And so you have one sampling, right? You have two big buckets of Skittles. You take multiple samples from each one, okay? So you take your hand, you stick it into one bucket, take it out once, take it out two, take it out three. There's your three samples from one bucket. Then you take your other bucket, take a handful, 1, 2, 3. You've got now 6 samples. Now you want to study diversity. So you can either look in each one of those samples, you count how many colors you have. In one, you have just blue and red. In another one, you have blue, red, and green. In another one, you have blue, red, and white. And so forth, right? You can either look at each one of those measurements, or you can say overall, okay, these are all the colors that I have. How do these colors represent the diversity between all of my samples? So either you look within each sample that you took, or between all samples. And between all samples will show you how diversity of colors, or diversity of bacteria in this example, separate your samples. Okay, so here are samples, each one has 3 colors, and these have 5 colors each. Okay, so that tells you, okay, so maybe those that have 5 or 4 or 6, they're all going to be here in this corner, and those that have 3, 2, or 1, they're going to be here. Okay, so that gives you that measurement. Of course, here we have a very simple characteristic, just color, but if there were thousands or maybe hundreds of these characteristics, right, now you get a little bit harder problem to separate them by diversity. So that is what NMDS is for. Okay, any other questions about what this analysis actually does and how it shows us the microbiome? How does it help us characterize the microbiome? Any other questions about it, or is it clear to everyone else? Okay, so now let's look at how this is associated with their behavior. Okay, so we have here the test results from their behavior. How do they actually test for different types of behavior? One of the ways to test it is to look at anxiety test. So anxiety test is measured by burying marbles. Maze behavior. Maze behavior is when you have a maze and you can study cognitive function. For example, how well does a mouse remember how to go through the maze, right? So you can do all kinds of tests to study behavior. And so here you can see this is your initial state of marbles, and here's how the patterns change between the two groups. Okay, the regular chow diet and the high-fat diet. So as a result of these tests, we can score them, right? And what do the scores show? This is elevated plus maze, time in open arms, so percentage of total test time. Chow diet, high-fat diet. So high-fat diet reduces the time in open arms. And here you have the percentage of marble burying. So this is the percentage of marbles buried. So you have chow diet, high-fat diet, right? So what you can see is that they perform, in a sense, better. I mean, they have less time wandering about, and they have more ability, they bury more marbles, which is associated with anxiety. Okay, so anxiety means that you are more stressed out and you are less calm and relaxed and exploratory. You are more focused, more anxious, more trying to get something done, more trying to, do these kinds of anxious behaviors. And so this is the outcome of this study. The study was able to show that it is not the diet itself, right? This is the important part of it. It is not the diet itself that you associate with the behavior. It's the actual microbiome. Because if you think about the way that they prepared the donor mice, these mice were prepared from a very early stage, right? They were just weeks old, and they were put on this diet for a long time. So that established a certain microbiome, because your microbiome gets established in the earliest stages of life. Once that microbiome was established, mice that had normal behavior before, they all of a sudden changed their behavior and they changed their cognitive function just um, uh, well, this is not the highest, this is what was given to them. So this is the initial, it was very organized and just, this is how many in total they have. And then they measure how many were buried. Okay, so what they were trying to show is that the microbiome composition and differences between these two groups actually indicate some relationship to, this gut-brain axis. Okay, so before we finish off today, let's see what kinds of data sets are available on NCBI about 16S metagenomic sequence. Okay, so how do we actually find this data? Okay, here we know that we want to look for Illumina sequencing. Okay, here you can actually look at different types of metagenomic studies, fecal microorganism, etc. Maybe actually let's switch it to bioproject. Okay, let's just look at the variety of topics that people apply metagenomic sequencing to when they study the microbiome. Impact of the gut microbiome and metabolome on the peripheral immune response in non-alcoholic fatty liver disease-related hepatocellular carcinoma. Okay, chicken gut metagenome, microbiome in feces of CKD patients. Molecular barcoding reveals bacteria of the stepomycetes as important members of root endophytes of apple plants grown in soils affected by apple replant disease. Eating behavior development in infants. The effects of oral colostrum care on very low birth weight infants. Rice microbiome. Okay, protocol for eDNA nano and phytoplankton. So yes, during spontaneous wine fermentation. Okay, so in terms of thinking about using the microbiome to study, different aspects of human disease and human health, development of infants, the conditions of soil and water, so you can study different environmental changes and how they affect the microbiome. You can study production of crops, so agricultural applications, a lot of different applications for microbiome. So we have about 10 minutes left, and I want to ask you, okay, so again, remember that one of your objectives here is to submit a project proposal. If you can take the next few minutes searching, okay, specifically in the bioproject database, find an interesting data set that you have found. So for example, if I find, you know, this, let's see what CKD stands for. Okay, it doesn't tell me. So maybe this is not a very good project for me to start with. So let me see, wine fermentation. Okay, maybe I am interested in the gut microbiome and how it relates to some kind of a disease. Meat processing environment, soils. Okay, so maybe I want to look at, let's say, acne. Okay, Chinese healthy and patients with acne, microbial diversity. So, or skin microbiome differences relate to the grade of acne vulgaris, or acne microbiome modulation, how skin cream can modulate microbiome and reduce acne symptoms, right? So all kinds of interesting, let's say, or maybe here, okay? So I'm really interested in this project, I want to study acne, okay? You have 10 minutes to find a project that is interesting to you personally, and just post a link. So I would take this URL from here. This would be the link to my data set. So I want to make sure that it has some data set, okay? And this data set is related to an interesting topic for you. So example, I will write, okay? So once you find it, let me know. I would put it like this. And post a link to the bio project. How do I know that this is actually metagenomic sequencing? If you look on SRA experiments, so for example here, okay, you can see what is the sequencing type. So Illumina, HiSeq, okay, metagenome, and you can see that it is metagenomic sequencing paired end. Okay, so for the next 10 minutes, please use this time to find a project, and then I have a couple more slides to conclude today's session. Okay, so we have Chandrani here. Let's take a look at that one. Soil and roots microbiome and resistome antibiotic exposure through irrigation water. Okay, so let's take a look. In this study, we assessed the effects of indirect exposure of antibiotics via irrigation water in microbiomes and rhizostomes composition of soil and lactica sativa, which is lettuce, in a controlled setting. We collected samples from soil and roots, and microbiome composition of all samples were analyzed by 16S ribosomal DNA amplicon sequencing. Okay, looks very interesting. And then let's see, we have overall 30 samples. Excellent. Let's see, Kylie. Expression data from kidney of 36-day-old PKD MHM rats. So let's see. Gender has strong impact on kidney performance. Female gender tends to be renal protective. PKD MHM is rat model for human polycystic kidney disease, PKD, caused by a mutation in gene ANKS6. PKD progresses faster in male rats. Microarrays to detect gender-dependent gene expression. Okay, so let's take a look at this one. Kylie, do you think this is microbiome? Okay, so to Kylie, this is not, right, not a microbiome. This is transcriptome or gene expression. So overall, it's an interesting data set, but it is not microbiome. So specifically for this, quick assignment today, it's not a good choice. So maybe look for another one. Okay, Megan Simonsen, H3K4ME3 modification profiling of Huntington's disease and control human postmortem prefrontal cortex brain samples. Okay, so again, look, data type is epigenomics, right? So whenever you look for H3K4ME3, that is a particular histone, right? Yep, so look specifically for microbiome. All right, Suparno, let's take a look. Suparno, determining the gut microbiota dependent impacts of anthocyanin-rich aronia berries on obese individuals of distinct inflammatory phenotypes. Overall goal of this project is to determine the inflammation-lowering impact of anthocyanin-rich aronia melancarpa berries. Inflammation is an underlying mechanism deriving the development of several disease, microorganisms, microbiome, host tissues, and immune cells residing in the gastrointestinal tract are a key source of pro-inflammatory signals. Okay, so this looks like raw sequence reads. Let's see what type of, 40 samples. Let's see what kind of samples are they. 16S ribosomal RNA, hypervariable region V4. Excellent. Okay. Yes, your interest and still microbiome specifically. Today, we're just looking for examples with microbiome. So Suparno, this is great. Primala, let's check what you have. Okay, microarray already tells us that this is probably transcriptome analysis. Yeah. So let's, okay, Sanjali, let's see. Pediatric CF lung microbiomes. Okay, so whenever you have abbreviations, it's always better to actually spell them out. What does this mean? This project, ECHO-CF, is a longitudinal study of the microbiome of pediatric cystic fibrosis. So CF stands for cystic fibrosis. Patients being cared for at Columbia University between February 2010 and June 2014. The samples, which were collected as part of routine care, are bronchoalveolar lavage, expectorated sputum, and oropharyngeal swabs. Okay. And let's see what kinds of, okay, so this is a huge project. It has over 800, and this is 16S sequencing of human airway samples. Okay, so this is a great project in terms of microbiome. Again, this was amplicon, right, metagenomic Illumina MySeq. So this is a perfect example, okay? Cystic fibrosis in pediatric patients. Okay, so let's say if you were interested to use this data, you would want to take a look at all the samples available and start determining what to do with them, right? Okay, so for those of you that are still searching, I hope this experience provides you with some insight into what else is out there. You could potentially use some of these data sets for your project. Okay, let's just quickly check Parimala, and that would be the last one. Okay, umbrella. So this is transcriptome analysis. So we have a project on mice. We have a project from the Human Microbiome Project. You've listed out great number of projects here, right? So you know how to search for them. I mean, there's too many to, you know, make a comprehensive example out of many of them. But I think that it should give you some insight into what is possible, right? So unfortunately, Parimala, this is still gene expression. This is not microbiome. Okay, so you want to include maybe microbiome. If you're interested in, you know, let's say immunological skin, or I don't know what your topic is, but whatever your topic is, let's say immunological microbiome, you want to search for that. Microbiome-based prediction of vaccine response in elderly patients. Very interesting. Fecal microbiota transplantation in subjects with irritable bowel syndrome, okay, mirrored by changes in gut microbiome. So, okay, so chronic spontaneous, let's see, I don't know what that is. Chronic spontaneous. It looks like you have some clinical data and some maybe specific genes that are associated with this. I don't know enough about this condition to know if there's a specific term that might be adjusted here. So what is chronic spontaneous urine? Okay, hives that are present at least, okay. So what if you look for this? Okay, so you can see in the human gut metagenome from the American gut, what we discussed, there's a specific sample that has this condition. Okay. Chronic urticaria and angioedema and allergic asthma. Okay, so this is self-reported conditions, but it looks like this is a part of the American Gut Microbiome Project. To the previous step, yeah, I just looked for the biosample. So there's just one sample. So maybe this is not enough to actually study microbiome data associated with that condition, but maybe, you know, it doesn't have to be a microbiome study, right? I'm just saying that this is just an example of how you find data. Okay, so to wrap this up, as we're out of time, I just wanted to kind of talk about where we will go next. So in our next session, we will talk about vaccines, antibiotics, and antiviral drugs. So we'll talk about how those are based on different types of metabolic pathways. We'll talk about some aspect of viral entry and replication, as well as immune system and evasion. And we'll talk about vaccines. What do they actually do? Okay. So Suparno, just to your question, you have a few projects right here, right? I mentioned that the other day. You can go to projects, and there should be some project examples. But also, we have shared with you some projects in the forum, I believe. Let's take a look. Okay, so we will post it right here. I see that it wasn't posted here, but we have quite a number of examples from other submissions and successful poster presentations. So we will definitely add it here today. Okay, so in our next session, we'll talk about vaccines, antibiotics, and antiviral drugs. I hope to see you all there. If you have the time, please go through some of those materials that were already discussed, metagenomics, genomics, some of the projects. It will give you a good sense of where to find data and how to structure it. And once you have those ideas, even a general sense of what you want to do with your data, with your project, start putting it into this form that we discussed for dependent project submission. And Dr. Vasudevan promised us that she will take a look at some of your project submissions for the weekend activity. So for those of you that have not submitted yet, please make sure to do so today. Today will be the last day to do that. Thanks again for joining, and hopefully I will see you again tomorrow. Okay, for those of you that want to do a group project, you can just specify that you will be doing this project with someone else. But yeah, you can submit it once, just one of you can submit, or if you want to submit both, that's also fine. All right, great. Thank you, everyone.